So hello everybody, my name is Marc Meunier. So in 2 minutes, I introduce myself. I work as a scientific consultant for Dassault Systemes and the Dassault Systemes brand, which I will briefly introduce here. So our Dassault's involvement in the project has been mostly around in the simulation groups, trying to characterize and understand mechanism of those nano materials, and they're finding some good descriptors to build some structure activity relationship, which then I will present. All right, so very briefly, let me introduce the system BioVia, what we mean by QNTR, Quantitative Nanotoxicity Relationship. I will talk about the data set of toxicity that were used at Otmar just showed before me this morning. Look at the nanomaterials and the atomistic models also that Nick was presenting. What type of descriptors we use, all the characteristics, size, shape, surface area, and dipole moment, and so forth that were used to build those SARs, and the models we built, and a briefly conclusion. So Dassault Systemes is a very large French, European actually, software company now, about 20,000 plus people, headquarters in the south of Paris.
Ah si, moi j'ai à peu près compris globalement, mais bon, c'est du Otmar après.
Yeah, I'm hearing
c'est plus
someone
des
here.
qu'autre chose.
Okay. Oui. So that's a system. That's a BioVia. The brand I represent really is about delivering software for the research in all type of industries, in chemicals and in pharmaceuticals. And today we will use some of the modeling and simulation software that BioVia provide to build our nanomaterials models, to compute descriptors, and also we'll use the tools to develop the relationship linking descriptors. So that is what we do in a QSAR, in a QSAR, quantitative structure activity relationship. We try to link a number of observations, so an inflammation response, and here we will play with the neutral cells, and link that to a set of independent variables, the X or so-called descriptors, right? And we want to correlate the Y's with the X's, and we can use all sort of function for that, and we try to really understand which functions or description of the molecules or the material here is responsible for the value of the Y's. So that's the first stage, and that would help us to understand the activity of a given material. But also it can be used then, and we hope as one of the aim of the project then to disseminate some ready-made SAR function that manufacturer could reuse in order to predict potential adverse effect of nanomaterials before they get synthesized. So by having the sets of descriptors for their given material, they could hopefully predict how likely the propensity for toxicity. So I mentioned the QNTR as a term I took from literature. Really, we look at the description of the bioactive nanomaterial, right? So the size, the shape, surface area, and so forth. So those are called the relevant descriptors. And in this project, we had about 60 nanomaterials that were selected, and that were mentioned previously this morning. And also, then you can use various machine learning and the robust QSAR modeling methods. So there, here represented, I believe, is a neural network, or with various layers you can use. We will use different techniques. We'll use some new recursive partitioning methods, but and also some genetic function that can be used. All right. So in terms of so the methods, and so we select the data sets, as I mentioned, and we take the data from our partners, and then play with the response. So we will use the log of the neutrals by cell count. We look at the chemistry. So I will show you how we build some relevant molecular models to describe those nanomaterials that we have. And then we calculate descriptors. Those are the, as I said, the physical properties of the material. And then there's a series of tools that we use to validate and build the predictive models. And so the machine learning here, as I say, the RP4S that we use. So that's the first large data set, and the second, so which I call a second data set, inflammation, is really derived from the first one as it's normalized by the experimental setup. So as a given, I will show you the sacrifice days and the dosage. Those are of course very relevant in the SARS. So we will normalize by those in order to get a single response, and then we will see how those descriptors then fare against this set of data. So in the first case we had 44 nanomaterials, and the second case we have 35 nanomaterials common. So I think Nick and Vlad already have presented this as a full list of nanomaterials, hopefully very well characterized. So they come also from previous European projects, and we have their description in terms of physical description, experimental data that we will use also for the building of the SAR. So the real materials are look a bit like that, whether the nanotubes, multi-wall or carbon blacks, and the nanoparticles, iron oxide, titanium, and so forth. So those are the real ones. And now we are building, based on these descriptions, we can build some molecular models, which hopefully are good representation of the real materials. However, those materials are very complex, and we will see that their size distribution, for example, is not always homogeneous, or we can have impurities, as just Luc mentioned before, and that can also have a large impact that we would not be captured in simple singular molecular model as such, but that's a start. Also, those nanomaterials are very often functionalized. Again, Luc presented the effect of hydroxyl or carboxylic acids very often. The silica also will have a various distribution of functions on top, and sometimes you have amines or ethylamines and so forth. So the molecular models that we built can also represent, have also a functionalization. So we wrote some script to automatically generate random distribution of such chemical functions on those nanoparticles or on the CNTs. Here you see a carboxylic acid modified surface of a carbon nanotubes. So those are molecular models, and so the toxicity data comes from the other partners, and we will use as a response the neutral cells. So that after curation, we have about 2,174 data points. This is a distribution just of the measurement. So that was very similar to what Otmar showed this morning about response by type. You can see is quickly just by looking at the data, the Y response is here, and by per nanomaterials type, which one has triggers the highest response already. And you see the graph on the right showing the thing, the distribution of the data set. Sorry, so we decided to go for using a log of the data because the data was response was spread a bit too much. across. The second set is based on the first one, and as I said, it's a normalization by to the inflammation. Here is a normalization considering the experimental setup, such as dosage and sacrifice day. So the data is a bit more homogeneous. We still have 2 points a bit that behaves a bit different, two response a bit different. So again, we in order to renormalize that data, we use the log of the inflammation response. So descriptors. So the first issue is that in a typical SAR study, we use small molecules, and they are pure as such. Nanomaterials is often a complex set of different chemistry, or if it's the same chemistry pure, then it might have a distribution. So think carbon nanotubes would have a distribution of length, or if it's a nanoparticle, you might have various sizes, and so you have a particular size distribution to take into account. So that is really tricky. But and also the experimental technique to characterize precisely the size, shape, length, and so forth of those nanomaterials is not always perfect. And we do have some, again, not experimental error as such, but deviation in the measurements. For the simulation computed descriptors, the literature reports redox potentials. That's very important for the nanoparticle, for the inorganic nanoparticles, and adsorption energy, band gap, and so forth, log P, and then we have the descriptors, as I said, from the protocol, from the experimental protocol. So 2 subsets of nanomaterial I mentioned. The first one is the 44 nanomaterials used in the Tox Dataset, and the second one we have 35. We didn't have all the data. So of course the experimental, so the materials are the same, common. So we use the same experimentally. We use the same descriptors. For the computed descriptors in lab, so the adsorption energy and protein, and I believe that is what Sasha Vlad will show later. How Nick mentioned the corresponding potential to look at the adsorption of those nanoparticles on given proteins and amino acids and so forth. So we will use that set of descriptors as well in our SAR. I mentioned descriptors, so the experimental, the physical chemical properties. The first question always is, do you take them when they are out of the manufacturing, or do you take them what they look like when they enter the body? And again, Nick mentioned the wrapping, the lipid, you have a protein coronas, you have all sorts of things. So if you think of the size of the nanoparticle, is it the pure size, or is it the size plus corona that you take into account? So that's really a challenge. As I mentioned before, the precise characterization of some of those physical properties is also a challenge. So we've used here mostly what was given in the list of the bill of material I mentioned before, the size and shape, the some sort of purities, functionalization, but we don't have, I didn't have data on PSD or crystal structures and coating and things like that. And the last class of descriptors is the experimental setup. We will see that is the most important. So we will use a second set in order to normalize with respect to those conditions. As mentioned, yes, computed descriptors, so from WP4, hydration energy, so that the attachment of water, and the bio-nano binding free energy, the Eads, but also surface area. And from my molecular model, I can also use all sets of computed descriptors. In terms of tools, so we use the Material Studio package for the building of the molecular model and some of the GFA SARs. and we use a pipeline pilot tools for the automating of the QSAR study using the RP4S. So the RP4S model were used as a technique to classify, if you wish, all the data in the various what's called trees and branches, and in the RP random forest, and you generate so quite recent techniques where you generate a very large ensemble of trees and classify your material, your sets of materials like that. So the first trial of QSAR here is just a bit of everything. So I used the entire set of log neutral ball cells as a response, and then all the descriptors that were available. And of course, you can see at the top here that most relevant for the variant is the are the protocol setups. So the sacrifice days, the dosage, of course. So it's more important in our case than the type of nanomaterial or the descriptors of the physical property of the material itself. Okay, so that's kind of self-explanatory. And then we have things like carbon counting. So that would describe if it's a large nanomaterial, carbon nanotube, sorry. So this with everything. And now if I add the computed descriptors from the simulation, from the adsorption energies, again, of course, the experimental setup still high on the list here, but then I start and a bit, and the surface areas, of course, extremely relevant. But then I start to see data showing, so the DPPC absorption. We'll see that that comes also when we will reduce the number of descriptors. We reduce them because we have a very high correlation between we use the entire sets of hundreds of descriptors, we start to have lots of correlation between our variables, our axes. So we do a correlation matrix, and then everything above 0.7 is removed. So if you think the surface area and the volume will be correlated, if you think the number of titanium and oxygen atoms as a descriptors each will be correlated, because we use TiO2 mostly. So so those are pretty obvious. Sometimes it's a bit less obvious, the correlation between them, but we try to remove as many as we had to. And for the Eads dataset, so there wasn't so much correlation with the rest, but between them, yes. So we only kept 5 descriptors for the adsorption energies, and so 1 for adsorption on protein, the TLR4, 2 on small molecules, there were small amino acids, and SER and serine, and also on lipids. And we kept DPPC adsorption as well, because that came across high in our first list. And so in this second data set, I had a go at a genetic function algorithm. So it's a different ML technique. I had a pretty decent correlation with the data, and it's also a way to reduce the number of descriptors. The GFA gives you can choose or limit the number of term in your equation. So here we see that the most relevant for our response is the length and the shape and the surface area. So all the volumetric, of course, as I was mentioned before by Otmar, I believe, is extremely important in the response, but also the nature here, carbon or titanium nanoparticles come. And then the adsorption of the DPPC, again, you see arrives. So the interaction with lipids seems as computed, seems to be a very important factor. And if I use now RP4S and my first ML technique again on this reduced data set with a reduced descriptor series, again, the surface area comes at the top, and then I have things like the adsorption on the lipid again, and also the lengths and diameters. So to conclude, and from the first data set, the large number of points, 2,000 plus points, Of course, experimental setup is a key to describe to predict and the response. So of course we need if you want to compare activities, we really need to make exact the same. Not only the nanomaterial has to be the same, but all the condition in which we test them has to be the same. It's kind of self-explanatory again. But the length of the carbon nanotubes, single wall or multi-wall, what the descriptors call atom count C, is highly correlated with our response. So basically, everything that looks like asbestos will trigger a very high response. And so if I excuse me, if I now normalize by those setups, then I find the surface area, most relevant length and diameter, so the shape again, and the adsorption of DPPC. So this interaction parameter seems to come up every time, and that's also explain the activity of the nanomaterial. And on this, I will conclude so we can have a little bit of a break.